What I want to do now is give you an update of what we've been doing. We've been working on this Rosette project for 1 year. Granted, it took us about 6 months to get the money, but we've sort of been working 1 year, half a year without money, half a year with money, So, but they expect everything done anyways. And so, to give you some sort of background on what we're doing, and that article gives you a background on it too, so you can read about it as well. But the components of the grant, there's 6 different components: diagnostics, monitoring, best management practices, breeding, which I'm very well involved in. That's what Ellen's helping me with. Economic analysis, and of course, information pipeline, which is to try to get the information out. Of course, the Facebook page is part of that. So we're moving along, and of course that article too. So we are trying to get things done. There's really an extensive network of collaborators here. It's just I lead it, and it's really fun because you get to talk with so many different people. But we have extension people in Texas, Oklahoma, Florida, and Tennessee working directly with the program, and indirectly we work with a lot of other people as well. Researchers in all sorts of fields, a lot of students and postdocs involved. Breeders, and this has been, to me, this was very important to work with the private breeders because they're the people who really push those varieties out. They have a lot more resources than I can to develop varieties, and so I wanted them to be involved at the beginning. And so I work with them directly and say, well, I need these crosses done. They make those crosses. They give me ideas what crosses should be done, and then we can continue with the work, and Ellen can look at them and what have you. So we're really working very closely with these people. A lot of different rose industry, 25 different companies, of course, the American Rose Society and a lot of other societies are involved, and hundreds of volunteers. And I'll talk a little more about the volunteers later, and you're always welcome to volunteer. And of course, a lot of universities, the USDA, of course, I mentioned the American Rose Society. But again, a lot of people involved in this project. But Rosette, I don't know if you've all seen it before. Hopefully you haven't. Oh, you have, sorry. It's funny because we don't have it here, and I don't want it, mind you. But, and Dallas is not too far away. I can go up there and see it. Anyway, so it's a viral disease. It's transmitted by a mite, very small, and we'll talk a little more about that. And of course, it's epidemic in the Midwest and Eastern USA. You don't see much in California, which is good, for them anyways. But little is known really about how to manage this. And it's an interesting disease from that point of view. There's 3 players in this epidemic. One, of course, is the pathogen, the virus. And we'll talk about what a virus is in a second. And the mite, it's an aerophoid mite. And it's really very small. It's not that big. It's what, 1/10 of an inch or something like that. It's really small. You need a magnifying glass to see it on the leaf. And it floats, so the way it goes from plant to plant, it can walk, but the plants aren't touching. It spreads its legs and it just floats in the air until it lands on something else. And so that's really how it spreads most of the time, long distance. And of course, multiflora, which was brought in here years and years ago into the U.S. It's native to Japan and China, and they use it as a conservation crop. So it has lots of fruit for the animals and hold the soil together and things like that. So they planted this all over the place, and it turned out it was well adapted, and it produces lots of seeds, so it keeps propagating itself. So it's sort of a noxious weed now, but it's there. And it's very susceptible to Rosette, and the mite loves it too. So it's this big body of a reservoir of disease that's everywhere, especially in the Midwest and the East. Not so much in California. And so here you can see where the multiflora is. That's the colored areas. And then there's a little in California, but we don't have the disease there, which is nice. Though originally the disease was in the mountains, in the Rocky Mountains. And actually, the mite is in California, but we haven't seen too much of the disease. But from the early 50s to recent days, it sort of just moved down into the West Coast. So it's been slowly moving. And years ago, 3-4 decades ago, they actually were thinking of using Rosette, and at that time they didn't really know what it was. It was a pathogen of some sort, as a biological control for multiflora. Well, multiflora is smarter than that, so it didn't really work, but I mean, you could infect multiflora with this. It was infected anyways. But so it's kind of interesting there. But this is what a virus is. And just, I want to give you some background to understand what a virus is. And I don't know if you do know, but what a virus does, outside its host, it can't do anything. It's got to be inside the host to function. And once it gets in the cell, and this would be a plant cell, you can see there's a little aphid here, just make that a little mite to transmit it. But once it gets in there, it goes into action, essentially takes over the cell and does its own thing. And so that's where it can reproduce. So it's integral to that plant once it's infected. It's systemic. You just can't separate it from the plant because it's there. And so that's what we're running up against is viruses are difficult to control because once they're in the plant, they aren't on the surface. You can't put a fungicide on and kill the surface stuff. It's inside. And so it makes it a little more challenging to control. And so we're working with a lot of people. These are my virology team, and these guys are great because they know. I can hardly explain what they try to do because it's just so beyond me from the chemistry point of view. But what they're trying to do is be able to diagnose the disease before you see it, actually. Because right now, we really can't do that. And this fellow is also kind of key here. This is Dr. Xanatakis from University of Arkansas. He's the one who first described this. And not that we didn't know the disease was there, we didn't know what was causing it. And it's only since 2011 we've known what it actually causes this disease. So, previously it was kind of hard to do anything because they didn't know what was causing it. Now we do. And so he's been very good about working with this as well. And so it was first described in 2011, and that's the picture of it. There's a little membrane and then some RNA there, which is your genetic material. Initially it was described with 4. Unfortunately, we found 3 more. So it's a little more complex than we thought. And so we're still learning a lot about this. And so, when people say, what do we know? Well, I think we know what we don't know more than anything. So we got a long ways to go. So we've figured out there's more than 4 RNAs. We have 7 RNAs. We're comparing different strains of the virus because it's anywhere from, the Rocky Mountains all the way to the East Coast. They could be different. So we're looking at that. And then we're trying to figure out how to diagnose it. How can we detect it in the plant? And of course, you have to sort of tag in on something like a DNA particle, genetic material, or some of the things that they produce with respect to proteins and things like that. And so this group of people that are working with me are doing that. And so currently, what we do, if we see the symptoms and see a mite, it's probably Rosette, and then we can take it and we can put it through a laboratory test and we can confirm that the virus is there. But it's a very expensive procedure at the moment. And we can only detect the virus if we see the symptoms. And we know there's virus before you see symptoms. I mean, that's the way it is. And so what we need to do and what this group of people are doing working on diagnostics is to develop an accurate, reliable, easy to use, quick, inexpensive test, something we can take out in the field. Something that, without a doubt, I can look at a plant and tell you if it has a virus or not. And this is essential for a lot of the work we do, even for the disease-resistant stuff, because even though it doesn't have symptoms, if it has a lot of virus, I don't want it. And we're finding out that the symptoms variety interaction is really quite amazing. There's some things that don't show many symptoms but have lots of virus. And so that's probably not good to be in your garden either, because if you have a susceptible one, well, everything else goes. And so, there's a lot of things that we need this diagnostics for. And what they're trying to do is develop a lateral flow device, which is kind of neat, because this is something you could actually take out in the field. And what it is you put your sample here, and then by capillary action, it moves over here, and this is a test line. So if in that test line it detects a virus, it changes color. And so nice and easy. Within 10 minutes, you have hopefully within 5 minutes, you have an answer. And so that's called a lateral flow device. Now, that's very similar to what they do with pregnancy tests. It's the same idea. course, detecting different chemicals. And so you can imagine being able to go out and take something like this out in the field and boom, you know if you have the virus. And so what they've done in the last year is they're developing new assays, and there's a lot of different approaches they're using. And they're reliable. Actually, they're more sensitive than the previous assay too. And this is they find one part out of, I don't even know how to say that number, but it's really quite accurate. So now they can detect the virus in a non-symptomatic plant, which is good because there's a lot of nurseries that don't want to ship, the virus out, but if they can't see it, they don't know if they have it or not. And so this is going to be very useful in detecting plants that have the virus that may not be showing symptoms yet. And so we got to do a lot more work and validation of some of these tests that we're developing, as well as further adapt them to the field. And so that's what we're hoping to get to. Now, we have a big project on monitoring. This is where we're using a lot of volunteers. And again, a lot of people are involved. These are all extension plant pathologists and people that deal with or have their own diagnostic labs. And these are plant pathologists, horticulturalists as well. Again, a lot of people involved in this type of work. And what they're doing is developing, of course, training materials, because all these volunteers have to be trained on how to recognize it properly, because there's a lot of confusion on the symptoms sometimes. In extreme cases, easy. But there's a lot of things you sort of go, well, that may or may not be, because there's a lot of things that can cause similar symptoms. So again, a lot of different materials are being developed, and we're almost there to be able to release some of these. And then a reporting app. Essentially, of course, we always have to do apps, right? That's what we do these days. It's got to be able to do it in your phone, otherwise it doesn't work. But anyway, so Dr. Ong is developing a reporting app so people can record the presence and severity of the virus or the symptoms, which varieties are on, of course the GPS so you know where, so we can map it out and how it's moving or where it is, and hopefully find varieties that don't get it overall. And so that's helped me in some of the breeding work as well. So hopefully by the spring of 2006 we'll have this app and we'll have all these volunteers energized and we'll start getting information in. So we're getting close to that. And so this is where we have volunteers in 31 of the 48 continental states, and hopefully we'll get the rest with time. And multiple people in every state. Of course, that's what you need. You need people throughout the state. But we do know that the symptoms vary with the rose cultivar, and this is a poster that was developed by Jen Olson up in Oklahoma, and she spent a summer taking thousands of pictures of symptoms and then checking them for virus. And so these are all the various symptoms that you can find for rose rosette. So some of it you wouldn't recognize as rose rosette. And then looking at best management practices, again, a lot of different people are involved here. And what they're trying to do is understand this relationship. So what you have the virus interacting with the rose. Of course, also interacting with the mite, which interacts with the rose. So it's not a straightforward situation. A lot of moving parts to try to understand. Try to understand the climatic effects and everything else, the genotype, the phenotype, transmission issues, the host range, and all that sort of stuff. So a lot of different things are going on and trying to understand this. And so they're looking at epidemiology and then just basically understand the mite and the virus biology, and then trying to figure out what can we do to control it in the field. Yeah, and this is both for gardeners and public gardens, as well as commercial people who have them in their selling lots, and the propagators and everything else. How do we control this thing? And so a lot of work is going on in Tennessee, and on a lot of this is Dr. Wyndham's working on this, and he's looking at planting design, effective barriers, plant spacing, how that affects the spread of the disease. So he has a lot of plots with various things. You can see these are the barriers. It's a grass that just sort of hopefully blocks that mite. It seems to be working for a little while. We don't know how long it works. So, and this is an area where there's lots of disease, and he's encouraging it too. Good plant pathologist loves disease. And they're looking at cultural control, the effect of roguing, whether you take the whole plant out or stems or what have you, how quickly you rogue. That can be very important. How can we train the landscapers? We're having some problems training landscapers because, again, there's confusing symptoms. And they don't always want to take plants out. That's the other aspect. So again, looking at the rate of disease spread, infections in adjacent plants, and he has 5 sites. He's going to be increasing this to a couple more sites in Tennessee and Alabama. So, and these are commercial sites that they're looking at and looking at weekly to see how things go. They're looking at various miticides to see which miticides may or may not work. And there's a lot of, there are suggestions on which miticides to use, but we're not sure if that really controls the disease yet. It might control the mite, we're not sure. So we got to do all this to make sure it actually works. And of course, get to the important part. I guess that's my part, right? Identify rosette resistance. And that's turning out to be quite challenging. And this article talks about that and what we've done to sort of get started. But if you look at sources of resistance, there are several species reported as sources of resistance. And these are Asian species. These are U.S. species. And one that's adapted or indigenous to Texas would be Rosette. And that seems to really have some resistance. Again, we're trying to confirm this with lots of different tests. And then there's things beyond, when you find something resistant, you have to try to figure out whether it's resistant to the virus or the mite. Because if the mite doesn't go on, it's going to be resistant anyways, right? But this is Rosa brachyata, which is a species from China, is resistant, turns out to be resistant to the mite, but it's very susceptible to the virus. And so if you look at, this is brachyata, you can see all the hairs there. And basically what's happening is the mite can't get close enough to the plant to feed well. It's not comfortable there, so it doesn't stay. It likes slicker things. You know, and this is not unusual in a lot of resistance to insects and mites. But the problem with working with species, they get big. These are some species hybrids. This is a Susceptible hybrid. You can see that this is actually 20 feet tall. And so it's huge. It's a rose tower and named appropriately. And so to start with that level of breeding and try to incorporate into a small shrub, you got a long ways ahead of you. So really, I'd rather start with cultivated things. And so we're making a big push to understand if there's any resistance among the cultivated rows. But we have tens of thousands of cultivars. We can't look at them all. And so we said, okay, well, where do we begin? We decided that we'd do observational information. Jen Olson up in Oklahoma and various people in Dallas and Tennessee, they had their observations, which ones were not getting it and which ones did. And so I started collecting all that information to see if there's any consistency in it. And so we had about 700 observations up to now, and I keep adding. We have plants classified as symptomatic, suspect, or no symptoms. And now some of them we actually have some virus information on them too. And so we did all this, and we came back to this. If you look at the class of rows here, and you see the various numbers with clear symptoms, about 219. This would only be the cultivated stuff. Things that were kind of suspected. They looked like they might have disease, but we aren't sure yet. And of course, the ones with no symptoms, we still have 40 without symptoms, which is good. Of course, now we have to plant them and try to infect them and kill them, right? So they're under assault. But you know, even with that, you know, we got quite a few. I didn't expect quite that many. And so we have to phenotype this, which we have to decide how resistant it is. And we're finding out there seems to be levels of resistance. So we have to measure the resistances of various people involved. Of course, Ellen and Jenkins, the other graduate student working on this project. So under test, we have about 40 different species accessions. Again, trying to confirm what people have reported to make sure we have the right accession of species. Because just because they say Palustris is resistant doesn't mean that all Palustris are resistant. It could be the one they looked at was resistant, and they probably only looked at one. And so that's always an issue. But we're looking at species reported resistant. We have 200 commercial varieties in our assay or our test. And of course, we've already identified 250 that are definitely susceptible, so we don't even put them in the trial anymore. So we've gone through at least 500 different things. We had another 50 or so to go. Probably end up to about 100 or more this year to put out there. But again, you get a wide range of diversity, all types of classes and types of roses. And I've talked to breeders and said, well, give me the most diverse stuff because I want to see if anything has any resistance. And of course, if they are, they're going to be used in breeding quite a bit, I suspect. So here's a shot of just some planting the germplasm out there in Tennessee, trying to get that resistance. And again, this is an isolated plot. They're augmenting as much as they can with Rosette. They introduce susceptible ones that are infected, try to get as much disease as possible. And again, just another shot of that. And so they're doing a lot of field screening. And what they do, they plant it, and then they get infected things and take shoots of that, put them on the uninfected, try to get it all infected with mites and disease. Again, we want to know if the plant can resist it, either by resisting the mite, or if the mite inserts it, it's resistant to the virus. And so various things. So once we see something that's resistant or tolerant, we're going to have to do grafting. And with grafting, if you graft a rose with an infected bud, the virus has direct path into that plant. So that's the way to test whether it's resistant to the virus. And hopefully it is, because that's what I would prefer rather than just resistant to the mite. So, but we have to distinguish between these different possibilities. We're also doing a lot of other phenotyping. Of course, we're phenotyping for flower types and Cercospora and black spot resistance and all this material. And this is our trials in Texas. Overton got planted this spring. We had problems with irrigation, so ours are still in pots. Hopefully this fall we'll get them planted and get those out. Again, we have 250, 300 different varieties we're trying to put out there. And then the genotyping. We talked about molecular markers. There's lots of different molecular markers out there. And this is the old-time molecular marker. Those are microsatellites. And again, working with Jekin and Ellen and Mandy. Of course, Dr. Klein works with us. She's the molecular side of it. I'm just a plant breeder. And what we're trying to do is develop, there's a new technology out here to really develop a lot of markers. Previously, when I've worked and made maps, that one map I showed you took us 2 years to make, and a lot of work. Hopefully with this type of approach, called genotyping by sequencing, or next generation sequencing using that technology, we can develop more markers, more tags on those chromosomes a lot quicker. And hopefully in 2 to 3 months, we can develop a map that's even better than what we could do in 2 years previously. And so that's what we're hoping to get. And this is what we've done. This is preliminary work. This is the old map where we had on this chromosome 17 markers. Now, on the new map, we have 125. The 17 took 2 years. This took 2 months, once we figured out a few details. And so we can go a lot quicker and get more markers, so we have a better chance of finding a marker that's associated with the trait that we're interested in. And then use that tag, let's say this cool 2295 1232, so we can follow that gene. They don't have really good names yet. We got to work on that. But anyways, so that's sort of the idea. And then, of course, working with a lot of breeders, and this is the range of breeders I work with. Actually, there's one more breeder who joined up, worked for Ball. Again, creating the rose crosses we need to do the analysis. And so this past year, we just got started. So we did about 3,000 pollinations, which, it's a good number. We did 1,800 with Palestris and Blanda, which are species that are thought to be resistant to the disease. Unfortunately, the results were not good there. We got very poor set and poor seedling recovery, so it might not be the way to go. That's what you learn. That's why working with species is sometimes very difficult, because they're not always compatible with the ones you want to cross with. Now, we did a lot of other crosses with Satidra, and they seem to be taking a lot better. Carolina derivatives, and among cultivated ones that we think are resistant. So we're just in the beginning stages here, and we should have, a couple thousand seedlings this coming year to look at. And then what we want to do is take all the phenotyping, essentially, is it resistant or not, put that with all the markers, and then there's some software we can put all that information together, and it tells us which markers are associated with the traits. And so that's sort of what we want to do, looking at pedigrees and all this information. So a lot of different things to do. We work with several people in Netherlands on the software and the analysis aspect of this as well. And then the final, or one of the other parts, not the final one, is the marketing and economic analysis. And we have three fellows, economists here working with us. Again, we want to understand how important this is from an economic point of view. Because we all say, oh, it's horrible, it's killed my plants. But when you talk to politicians, they say, well, how much is it worth? Well, it's a good question. And so, usually scientists say, we don't care, it's just horrible. But, so these guys are going to tell us how much it's worth. We hope anyway. And they're doing some interesting things. You see, he's hooked up with electrodes, and they're trying to measure his brain waves to see if he's sane, I guess. I'm not sure what. But they're going to look at consumer preference and a bunch of different things, again, to quantify the relative importance of various traits, disease resistance being one, but they're going to go beyond that to sort of understand the rose as an economic crop. Looking at barriers in the marketplace to get these roses out. Once we develop resistant ones, what do we have to overcome? What do we need to really make them worthwhile? Because we can get something resistant, but if it doesn't have everything else, no one's going to plant it. I can tell you don't want to plant Rosa satidra. It's resistant, but it flowers once a year and it just takes over your whole yard. You don't really want that, I suspect. And then look at the socioeconomic impacts of rose rosette. I'm not quite sure how they do this, but they tell me they can. I believe them. And so they're using a lot of the biometrics here. They're looking at brain waves and galvanic electroconductance to see how emotional you are. They tell me all this works. Read your face. So be careful when they test you. They're going to read your face too. And all sort of visual attention behavior, all sorts of behavioral things to understand what's going on in your mind. And how do you sell you more, basically. And of course, we want to sell you more roses. So this is good. And then we have the information pipeline. These are extension people that are working with us in developing materials to get things, the information out. And of course, they're helping with the Facebook pages. There's a Twitter account, though I don't know how to use Twitter yet, so I don't know the account. Writing publications, extension bulletins, all that sort of stuff. And of course, getting students out to talk about it and tell it at the American Society of Horticultural Sciences meetings. As, of course, we had several other students go to, always talking about various things that we do. We're getting press releases out. We're developing online resources for training and information and various things. So we're trying to get all this out. After a year, we're getting close to start pushing information out. And of course, again, the grant is complex. There's a lot of different things we're trying to do. And this first year has been sort of, trying to push everyone into it, and let's just get things established. And the diagnostics people have gotten a lot done because they do everything in the lab. It's a lot easier. If you get in field stuff, we're just trying to get things planted. It takes a while to get going. But we're getting there, and next year is going to be really exciting. How about current management recommendations? I'm sure you're all interested, right? There's two ways to do it. You either exclude the virus or reduce the mite spread and mite populations. That's the two approaches you have. Okay, so obviously only plant roses that don't have rose rosette. Simple. Well, if you could tell. If it's symptoms, you can tell. If it's no symptoms, well, you really can't. And so one of the things we're doing is trying to figure out whether the infected plants are being shipped around and see if that's a major issue or not. So that's one of the things, because we haven't had a way to tell yet. Eliminate rose rosette infected roses from 100 meters of your garden. Well, if your neighbor has them, well, you're going to have to talk to them before you rip them out. But it's good suggestion, because that you don't want anything near your nice roses. I mean, there's no doubts there. Monitor weekly and eliminate rogue out plants with symptoms. So I missed a word there. But they're talking about monitoring weekly, and that's a hard thing to do. And in South Carolina, they're doing this. No, not South. In Tennessee, they have a public garden, where they monitor weekly, and every week, if something's showing any symptoms, they think it might be rose rosette, they take it out. And by doing this, and this is a very high pressure area, there's a lot of rose rosette in the area, they have to replace maybe, he said, 3 to 4% of the roses every year. But he said, that's not a problem. I mean, you're talking about 1,000 roses in this garden. So taking out 30 or 40, not a big deal. And that way it doesn't get started. And so you have a real nice rose garden anyways. He says, you know, he does this, and the curator probably doesn't even notice he's taking things out. So, you know, it works to do it that, but they're monitoring weekly. And so that's key to get them out quick. If you want to reduce the mite spread, you don't plant them real close together. A lot of these commercial plantings are so close together, I mean, the mites just walk from one to another. You know, they don't even have to fly. They just walk. If you remove it, you need to bag it. And the problem is, if you don't bag it, those mites fly, and they stay in your garden. So you have to take it and actually put it in a plastic bag and get it out before those mites spread. Because once that plant starts desiccating, those mites go up and look for a wind or a breeze to take off. Do not use blowers. And you'd be amazed how many people use blowers to clean out their roses or any landscape situation. If you use a blower, well, you're blowing the mites all over the place too. And so that can be an issue. Do not go from an infested garden to another garden. And you say, well, why not? You know, it's virus. It's not going to attack me. Well, the mites are going to be on you. That's key. That's not the virus per se. That's not going to be carried on your hands or anything, but the mites are going to be in your clothes or what have you. And you go to another garden and a gift that they don't want. Prune heavily in the winter. You do this, you eliminate mites because they overwinter on the canes. So if you eliminate it in the winter time so you don't have any mites or less mites to start with, you're in better shape. And then dormant or summer oil may help. It seems like oil does decrease your mite populations. And there's certain oils you can apply that are safe and work fairly well. And so that's sort of the current situation. Yes?
How long do they live not on a rose, but say on your clothes or on a shovel or something?
They will, as far as we know, and this is still a little guesswork, 1 to 2 days is what they're going to survive. So it's in terms of a couple days rather than hours. And of course, or months, yeah. No, it's not months either. But, you know, if they're on your clothes and they want to fly, well, it's easy for them to fly too, you know. So, and you know, the worst thing I can think of is a landscape person pruning out these roses, putting in the back of their pickup truck and taking off.
Without bagging.
Without bagging. I'm sure that happened, but nobody knew. You know, so it's not their fault yet. They don't know. They're beginning to learn, and I guarantee you there's a lot of interest in the landscape industry to be able to manage this very, very quickly. Yeah.
If the plant becomes infected and you pull it out, is the soil under it?
No, the virus won't be in the soil. Though you have to be careful. And some people say, oh, you should take all the roots out. Well, probably not, because again, the virus may be in the roots, but it's going to die with the roots. So it's not going to go from a dead root or a dying root into a plant. But if you leave part of the stem there and it shoots up again, you're going to have it. So you have to make sure you get all the stem tissue out so there's no more shoots coming up. But you don't necessarily have to take all the roots out. Okay? So that's important too.
When you said prune heavily in the winter, do you mean around Valentine's Day or winter?
Yeah. I mean, you can do it before things start growing again, because the mites won't be very active when it's cold, because they're cold-blooded, so they just hunker down. All right, well, again, we have lots of collaborators. And of course, these are some of my students. And this is really the reason that makes it so much fun to work with students. They're really, and I do appreciate your support of students. It's been wonderful to have this scholarship available. But you can see they're all still smiling. This was in August up in Mansfield, after being out there all day looking at roses. And it was, 95 to 100, somewhere in that range. And so, you know, to be able to smile after that, that's amazing. And so I think they were happy because they're going home too, but you know, whatever. And here's a group that went to the national meetings as well. Of course, Ellen's there, and then 4 other graduate students went. So we had good representation at the national meetings as well.